Full schedule.
Okay.
Because we have a full schedule, I think we're going to go ahead and begin. And my name is Arne Kumigai. For those of you who don't know, I'm Vice Chair of Education for the Department of Medicine. And I have the absolute delight in hosting the citywide medical grand rounds. This grand rounds in particular is noteworthy because we are celebrating the winner of the Canada Gardner-Whiteman Award for 2021. Dr. Elizabeth Eisenhauer is the 2021 Gardner Laureate. She hails from Queen's University and is well known to many people on this call. Received her MD as well as postgraduate training in internal medicine and hematology from Queen's. And then from 1982 to 2012, she was the director of investigational new drug program at the Canadian Cancer Trials Group, and has led over 170 clinical trials on new cancer therapies. From 2012 to 2017, she was professor and head of the Department of Oncology at Queen's University in the cancer program, medical director at Kingston Health Sciences Center in Kingston. She is currently a professor emerita, and we're debating on how active her emerita status was, which appears to be extremely active at Queen's University. And since then, as an indication of this, since January of 2018, so she was pulled out of so-called retirement very, very quickly. She's been the head of the innovation lead at the Kingston Health Sciences Center. For her contributions in cancer research and her many roles, leadership roles in Canada and internationally, she is awarded the O. Harold Warwick Prize of the National Cancer Institute of Canada, now the Canadian Cancer Society. The Society of Gynecologic Oncology of Canada Presidential Medal, the Queen Elizabeth II Diamond Jubilee Medal as well. In 2017, she received the Canadian Cancer Research Alliance Award for exceptional leadership in cancer research. And later that year, she was named an officer of the Order of Canada. In 2021, as I just mentioned, she was honored with the Canadian Gardner-Whiteman Award for contributions to clinical cancer research and care. So this is somebody who has tremendous background and has made tremendous contributions in clinical cancer research and delivery. Dr. Eisenhower's talk today is titled Improved Cancer Outcomes, but Miles to Go Before We Sleep, Personal Reflections on 35 Years of Cancer Research. And with that, I would like to introduce Dr. Elizabeth Eisenhauer with our congratulations. Elizabeth, please.
Thank you very much. Thank you, Arno. And it is great to, at least virtually, see so many of my friends and colleagues from the years. I'm really privileged to speak to you today, taking you on a little bit of a journey of what I have been doing and how I have grown over 35 years in cancer research, and also bring to your attention, not only have we improved cancer outcomes quite dramatically in some instances, but there's still a very long way to go before we can rest. So let's go back and set some context. Back in 1982, when my work in this field began, what was happening in the world? Oh, sorry, I have to go through my disclosure slide here as well. Non-relevant, and the objectives were circulated in advance. So back to 1982, when my work started, a number of momentous things going on. In Canada, we finally patriated the Constitution from Britain in April of that year. The desktop computer was the machine of the year. There were some things happening medically. The acquired immunodeficiency syndrome was being described, increasingly garnering interest, research, and publications as a new disease. And in the cancer field, a lot of attention being paid to this couple of researchers, clinical and mathematical, from Vancouver, Goldie and Coldman, who developed a mathematical model for increasing sensitivity of tumors to drugs by giving them alternately. And that in turn led to some clinical trials, as you can see in the top left. evaluating this approach in the treatment of particularly lymphomas. But also at that time, Michael Jackson released his Thriller album, and a young man from New York City clearly was starting to exhibit interest in touching things presidential, sadly, some might say. On a personal level, July of that year, I was the mother of a 3-month-old daughter, Nicole, and a 2-year-old, Danielle. So that was where I was. Meanwhile, cancer's story, of course, goes back millennia, as nicely laid out in this lovely, almost a novel, a biography of cancer by Siddhartha Mukherjee. One of the first documented cases of breast cancer written about was in 500 BC, when the Persian queen Atossa, apparently by the description in the text, had a tumor in her breast, which afterwards spread and killed her. Despite how long we've known about cancer, it took hundreds or thousands of years before any meaningful treatments came. And there were limited benefits really from any sort of treatment offered until the late 19th, early 20th century, at which time surgery was enabled by the discovery of safe anesthesia, and sterile techniques were developed for surgical procedures. Although radiation was discovered around this time, it wasn't really used therapeutically for another half century. And many patients with cancer died within a few months to years. And in fact, in 1947, when the Canadian Cancer Society created the National Cancer Institute of Canada to undertake research into the causes and treatment of cancer, 5-year net survival rates for all cancers at that time was only 33%. Things began to accelerate and change in the 40s through the 80s, with therapeutic radiation becoming possible, with better understanding of physics, radiation dosing, safety. New imaging technologies made it possible to appropriately stage cancers and plan treatment. And the first so-called chemotherapy drugs had been discovered, many through the US National Cancer Institute drug development and screening programs. And some examples are shown here. And of course, there's a link between one of these, vincristine, an extract or derived from the Madagascar periwinkle, which had a history related to some discovery work done at Western University in Ontario by Robert Noble. Now, 1987 is the earliest you can find online cancer statistics for Canada. So this was a few years after I had started my job. But even at that time, you can see that the common solid tumors in leukemia pretty uniformly, with the exception of breast cancer, had 5-year net survivals below 50%. The 1980s were a busy time in cancer biology and clinical trials arenas. Big changes were afoot. So in the laboratory side of cancer research, there were new technologies, techniques, and the development of cancer models, which could be used by scientists to begin unmasking those fundamental biologic changes in cancer cells, and foreshadowed what might transpire 2 decades later within the treatment sphere. And in the clinic, Canada and many other countries began to create cancer clinical trials cooperative groups or networks to conduct multi-center academic-driven research studies intended to accelerate the pace of the development of effective new cancer treatment. In 1980, with funds from the Canadian Cancer Society, the National Cancer Institute of Canada launched a Canadian Clinical Trials Group, the so-called NCIC-CTG, to coordinate and conduct cooperative clinical trials to improve cancer outcomes. And the trials that it was working on at that time actually had begun independently of a formal group being created. Trials started in the 70s by individuals actually from Princess Margaret Hospital, such as Ian Quirt and Danny Bergsegel. And these trials really needed a home and some standing infrastructure that would allow increasing work to happen in Canada. The inaugural director of the NCIC-CTG was Joe Pater, whom many of you know. He's an oncologist and scientist and epidemiologist based at Queen's. And it was actually because he was already a member of Queen's faculty that the central office of the CTG ended up being placed there. And in 1982, as was mentioned earlier, I joined this NCIC-CTG as the inaugural director of the Investigational New Drug Program. And here's a photo from that, well, probably 84. You can tell by the shoulder pads, we're definitely in the 80s. So the mission of the NCIC-CTG at that time and to this day has been to develop and conduct trials aimed at improving the treatment and prevention of cancer with the goal of reducing morbidity and mortality. To do this, there were three interlocking key components. The first was a coming together of scientific and clinical leaders across the country from which the ideas for the appropriate trials were developed. The second was the bringing together a network of cancer centers and hospitals where the patients who were enrolled in those trials were treated. And the third was to set up this central statistical and coordination center where protocol development, ethics and regulatory oversight, physicians and statisticians worked to ensure the trial conduct was appropriate and analyses were undertaken. And all of these linked as a triumvirate, if you will, is what the NCIC-CTG was and is. And as many of you know, in 2016, the group was renamed the Canadian Cancer Trials Group, or the CCTG. Now let's go back to the beginning of the IND program. In the 70s and 80s, most new cancer drugs were identified and developed by the US NCI, Developmental Therapeutics Program. Important and perhaps somewhat ironic to note that pharmaceutical industry was not very interested in cancer drug development as a therapeutic area. It was felt to be high risk and not provide enough return on investment to be profitable. So put that in the back of your mind, and we will return to that later in the talk. At the time, Canadian investigators, and there were a few interested in new drugs, as well as their patients, wanted the opportunity to be part of these early phase trials where promising new agents were evaluated. And these were awarded to groups based on letters of intent submitted to the NCI. And in 81, NCIC-CTG set up a new drug committee chaired by Brian Weinerman, who looked at why we weren't that successful in getting very many studies in Canada. And it was really felt to be because we weren't on top of what was happening in the pipeline in the NCI US. And we really needed to create a program and hire a clinician to run it who would be responsible for connecting with the US National Cancer Institute Drug Therapeutics program and identifying promising new agents, connecting with Canadian colleagues, and getting letters of intent submitted so that we could be part of this growing area of interest. So at that time, that was me, that was the junior oncologist hired. Actually, it was Danny Bergsagel who suggested to Joe Pater that it may be something I could do for a couple of years. What were my qualifications? Well, pretty limited. I had completed a training program in hematology. There was not yet a formal medical oncology training program in Canada. I was once actually enrolled as a subject on a clinical trial, and that is a longer story for another day over a glass of wine. I played violin in the local symphony orchestra, so I did know a little bit about teamwork, but pretty much I had nothing else in terms of training except I was keen and I wanted to do clinical research. On the other hand, at that time in Canada, few had any experience in training in the area of new cancer drug development. So my first lesson today is expanding your horizons to do things in which you do not yet have expertise can certainly lead you to very interesting places. So in the early years of the 80s, we began by focusing primarily on phase 2 studies. Those are trials designed to screen new drugs for evidence of activity in patients. And as the scientists in the room will know, despite the discovery of the first oncogenes in the 70s, and what that might mean for future research, almost all of the drugs we were looking at were probably best classified as cytotoxic agents. We definitely had a few lemons in the first year of the program, but also some kind of interesting results. So on this slide, you'll see IND 12. Cleverly, we adopted a nomenclature system whereby each successive trial got a next higher number. So IND 12 was a phase 2 study of CBDCA, which is a drug now known as carboplatin in ovarian cancer. And this was the first trial of carboplatin in ovarian Canada. And we were pleased to see that in recurrent disease patients, we saw about 28% respond. And later, the CCTG went on to study this drug frontline. And in time, the results of that and other studies led to this drug now being part of the standard of care in frontline ovary up till this day. Another interesting trial was that of deoxycoframicin in a rare form of leukemia called hairy cell leukemia. And this also was interesting because it was one of the examples of research coming from labs in Canada moving into the clinic through the clinical trials group. So James Johnson, who's a hematologist in Winnipeg, was evaluating DCF and other drugs in some cultures in his lab, and it looked really promising. So somehow we got drug supply, we got Health Canada approval, and we started to evaluate it on a 3-week, a weekly ×3 schedule. And the overall response rate in that trial was 100%, with 90% or approximately 90% of patients having complete remissions. And interestingly, I was a practicing hematologist at that time. And the very first patient enrolled in this trial was a gentleman who had, I guess, the good fortune to be diagnosed with hairy cell leukemia just as this trial was opening. And he had a massive spleen filling his left upper quadrant. He was cytopenic. He had his marrow filled with hairy cell. And after only 3 weekly doses, when I saw him, his spleen was no longer palpable and his marrow was showing already dramatic regression. And I can tell you that one early experience in my career was extremely exciting. There's just really nothing more exciting than participating in research as an investigator or as a patient that has such a dramatic impact on someone's life. The 90s can be called the taxane decade, I think. Taxol, which was its original generic name, later adopted as its trade name with the paclitaxel as the generic, was an extract from the bark of the Western yew found to be active in the NCI screens in the 1960s. At the time, the US Department of Agriculture was sending thousands of natural product samples to the NCI to screen in their cancer screening systems. It was not particularly interesting to pursue. People thought it was a tubule poison, so we had some of those, why bother? Until a novel mechanism of action was first described by Dr. Susan Horowitz in the 70s, when it was identified as being something that stabilized microtubules and didn't prevent them forming. So it got into clinical trials in the 80s, but had significant cardiac toxicity problems, severe bradycardia and other arrhythmias. Severe hypersensitivity reactions started to appear, which were presumed to be related to the cremophore used in formulation. But because some evidence of activity had been seen in ovarian and breast cancer, there was work undertaken to prolong the infusion, to add pre-medication, and to see if we could make the drug more tolerable by these maneuvers. And this brought my first foray into not just Taxol trials, but ovarian cancer trials, because in talking with some colleagues, we decided to do a study that would examine different approaches to administering this drug, looking at different schedules and different doses in a bifactorial randomized trial. And this also was the first international randomized trial experience I had, because about a dozen European centers joined with the CCTG to undertake OV9. And the findings of this trial really ended up changing our thinking and our practice around Taxol. The shorter infusion, remember, it had been lengthened as a way of making it safer, but surprisingly, the shorter infusion given with pre-medication ended up actually being safer. It produced less myelosuppression. and it didn't lose any of its efficacy. And indeed, the higher dose seemed to be more effective. This not only changed practice, but led to two important subsequent developments. The first was a pretty obvious one. Canada had led this trial in recurrent disease, and it was obvious we now needed to move it into frontline. So our European partners, together with us, designed an international or an intergroup trial of frontline Taxol platinum compared to the then standard of care. And we showed that despite a 50% crossover rate, we were able to improve overall survival in ovarian cancer by a median of about a year. And this Taxol platinum, carboplatin, in fact, combination remains the backbone of frontline ovarian cancer therapy. So that was one important outcome of this initial work. But the other one was it whetted our appetite for thinking about doing intergroup collaborations across the pond and north-south in North America. And we were one of the founding members, as a result of this experience, of the Gynecologic Cancer Intergroup, which is one of the first formal intergroup organizations in the cancer space, started in about 1997. And the goal of the Gynecologic Cancer Intergroup is to bring together representatives from cooperative groups around the world interested in gynecologic cancers to develop consensus understanding of the best standard treatments, and also to define where there were gaps in knowledge, develop research questions, and collaborate on trials together. And this has led to, of course, lots of collaborations over the years between Canada and elsewhere. There are now 33 groups around the world, part of the GCIG, and it's also led to lifelong friendships. And you can see a very young me there with Martine Picard back in '93. In the '90s, taxanes were now everywhere. It was the new great hope in cancer, dozens of trials ongoing. And in addition, it took for this drug to get into practice almost for the first analog to appear. Again, a big change from today when analogs appear simultaneously across the board. First analog was docetaxel, a semi-synthetic taxane from the European U. And it started phase 1 trials with a theoretical advantage that it wouldn't need pre-medication because it was not formulated in Crema-4. And because of our success in Canada from OV9, the NCIC-CTG and the IND program obtained this agent for 5 phase 2 studies. And I'm just highlighting probably the most exciting of these here. It was a breast cancer trial that Maureen Trudeau from Sunnybrook led. And by the 10th patient, we knew 2 things with certainty. The drug was extremely active. 7 of the first 10 patients had very rapid and sustained partial responses, and almost all of them also had hypersensitivity reactions. And so this drug, docetaxel or Taxotere, went on to become part of the standard of care of breast cancer, and pre-meds are given when it's administered. By the late 90s, change was definitely in the air. Targeted drugs were emerging. There were discussion about new endpoints in trial design, and professionally, my life was changing as well. So this new wave of first targeted drugs, it's important before I talk about them to point out that all cytotoxic chemotherapy agents actually do have molecular targets. But this new wave and this use of targeted therapy nomenclature really was focused on agents that were intended to inhibit or affect the signaling pathways and receptors thought to be the root cause or the drivers of uncontrolled cancer cell growth. And this little diagram of the entire signaling network of a yeast cell conveys some of the complexity here. And despite the complexity, there were a few specific targets or receptors and pathways that floated and bubbled to the top as being repeatedly abnormal across many cancers. Many new agents emerged, and some had quite stunning success. But it's important to also acknowledge what we were thinking the world would be like at this time when these new agents came into the clinic. First of all, it was felt they needed to be exquisitely specific. We couldn't have a drug that hit a number of different molecules. They had to be highly specific. Secondly, they needed to be given continuously to work because it was imagined that they would not cause tumor regression, just slow or stabilize growth. It was felt that they would likely be non-toxic and change cancer into a chronic disease. And that furthermore, because we were targeting something that we knew was, for the most part in the early days, expressed on cell surface receptors, we should be able to easily define biomarkers to select patients and identify who best to treat. That's what we thought then. And about this time, I was invited to give a talk at a new drug development meeting in Amsterdam to discuss my views or my predictions around early drug development and how it would change. And what I said, and I'm on the record in this paper as saying it, was that instead of looking at toxicity to guide dosing in phase 1, we would be making our recommendations based on optimal target inhibition. And instead of relying on tumor shrinkage and responses to identify active agents in the phase 2 portion of drug development, we instead would be looking at prolonged disease stabilization and require randomized designs. Needless to say, the IND program did a lot of clinical trials with targeted drugs in the next few years. And I show here a long list of both targets and the agents' names. But most of those ended up being negative with some exceptions. And I mean negative in the tumor types we evaluated them in our phase 2 programs. The exceptions were the proteasome inhibitor bortezomib, which showed activity in mantle cell lymphoma and in Waldenstrom's in our hands, and the RAF inhibitor sorafenib, which we studied in AML with Mike Crump chairing a trial, and it went on to be further explored in that setting for people with FLT3-ITD mutations. At the same time, the clinical trials group as a whole was undertaking phase 3 trials, and we had 2 signal successes at that time, both with EGFR inhibitors, the first in colorectal cancer with cetuximab, and the second in non-small cell lung cancer with erlotinib, and Francis Shepard was a leader of that trial. And both of those led to approvals in Canada, in the US and elsewhere for the use of those drugs in those settings. And both of them also downstream from their clinical work identified biomarkers for patient selection. By 2005, I think we could say we had a bit of a mixed response to how targeted therapy was doing. There were definitely striking and game-changing results in some settings, such as the imatinib CML story. There were some less visually dramatic, but nevertheless important and practice-changing observations, such as the HER2 inhibitor trastuzumab, given as adjuvant treatment in breast cancer. Many were negative. This is a gefitinib trial in non-small cell lung cancer. And some were even worse than negative. Here's an example of a MET inhibitor with erlotinib in non-small cell lung cancer, where the combination arm did worse than the standard. So the bloom on the rose of targeted therapy, I say, was fading slightly. Maybe better said that some realism was setting in. Not everything was going to be in the imatinib and CML. It didn't mean we were going to throw the baby out with the bathwater, but we needed to take off the, shall I say, rose-tinted glasses about targeted therapy being a panacea. In addition, the clinical results of these agents proved that essentially almost all my 1998 predictions were wrong. First of all, these drugs were toxic. They were just toxic in a different way than standard chemotherapy. Secondly, looking for optimal dosing based on target inhibition was challenging and frankly, sometimes wrong. targeted drugs actually did cause tumor shrinkage. And in a paper that I did with one of my fellows, Robert L. Maraghi, we looked across a whole range of targeted drugs and tried to correlate how well they did in causing response rates as single agents with how well they did in improving survival when they got to phase 3. And there seemed to be a very significant correlation. In other words, if your targeted drug caused single agent response rates of 20 to 30%, it almost certainly, not guaranteed, but almost certainly would have an impact in the randomized phase 3 setting. And finally, biomarkers, yes, indeed, they were important, but certainly much harder to identify, validate, figure out than everyone thought. So this is humbling. Predictions based only on imperfect knowledge of biology from the lab setting may be wrong. And so we really need to be open-minded and evaluate our hypotheses in the clinical setting and focus on the effects seen in clinical trials to actually further our knowledge. As all of this was happening, I was starting to get involved in leadership in a number of organizations. I'm not going to talk too much about them here, except to say that life was already pretty busy. And I was often asked, why was I doing this? Especially from my family. And I had a couple of reasons. First of all, I felt quite strongly that clinicians needed to be part of discussions about cancer research directions in general. We need to know, especially those of us doing drug development, where is the basic science going? How do we start to anticipate what the questions of the next 5, 10 years will be? How can we help basic science by integrating what we know from the clinic to ensure questions relevant to patients are being evaluated and addressed? I also think clinicians need to be engaged in solving health system problems. We have to not just serve the individual patient before us, but society writ large. We need to find ways to give back, teaching the next generation. Here's a group of early investigators in Switzerland in the FLIMS course for clinical cancer research methods from about 15 years ago. And we also need to support and help grow those organizations that in turn are supporting our research. And finally, and maybe this is for me the most rewarding, is that it gets you to meet lots of interesting and new people, and new and lifelong friends can be made. So moving more proximally to today, between 2005 and 2010, many new targets and drugs were being discovered. The genomic, the omic revolution, the rise of the mantra of precision medicine, and trials focused on this were coming our way, as were anti-angiogenesis drugs. I'm also going to talk a little bit about what I call trial endpoint regression and moving from cancer research into more cancer control and policy. So the brilliant Judah Folkman really led the way to describe the role that angiogenesis, or new blood vessel formation, had in promoting and sustaining tumor growth, and also in identifying inhibitors of this process. So expectations were high when the first one of these drugs, called endostatin, went into clinical trials. And there were whispers in the corridor. And I remember, some of my colleagues in Toronto, including Bob Kerbel, who was also involved in this field, saying that at that time, is it possible that in 6 months we'll know whether a cure to cancer was finally in sight? So naturally, the IND program, we were involved in some of the early trials of these angiogenesis inhibitors. I'm only going to talk about one at the moment. This one, AZD2171, or cediranib, was a vascular endothelial growth factor receptor tyrosine kinase inhibitor. And we did phase 1-2 trials in the IND program with this. And that led to 2 randomized trials in lung and in ovary. And the lung study is negative, but the ovary study, which was done in which we collaborated with the UK, which led the trial as a data center, showed that the cediranib combination in relapsed disease with chemotherapy significantly improved not just progression-free survival, but there was definitely a trend to overall survival improvement, which didn't, as you see there, reach statistical significance. But these observations have led to ongoing research with this drug in other settings. The anti-angiogenesis wave of trials made the taxane wave look kind of like a little tiny flutter. At one point, there were more than 1,000 trials ongoing with the VEGF inhibitor bevacizumab alone. This was one of the first examples that I could remember where you had multiple analogs, if you will, simultaneously in development, all competing for patients with the same ideas. I would say, despite all of these studies, angiogenesis inhibitors have been disappointing. They have only rarely had a meaningful impact on cancer survival. And there are some lessons to take from this. One is around the preclinical models. So as many of you know, preclinical tumor models actually have to rely on pretty dramatic tumor growth rates to get established and to evaluate treatments. And thus they were very reliant on angiogenesis when you think about it. And if you look from this ancient, I won't say too ancient, but ancient to some of you young people in the room, publication from Ian Tannock, if you look at this on this log scale of the growth rates and doubling times for a transplantable mouse tumor versus a human breast cancer, you could imagine that angiogenesis would be really important for that mouse tumor, but maybe not such a big deal for the breast cancer. So an important lesson there. And the second lesson is when a drug or drug class has limited activity, you cannot turn it into something that is dramatically effective by just doing hundreds or thousands of trials. I refer to this little topic as trial endpoint regression because, as you will recall, those predictions I made about looking at inhibition of tumor growth as something that targeted drugs would do were largely proven false. Nevertheless, the primary endpoints in cancer trials started to shift. In randomized trials, progression-free survival, or time to tumor growth, was and continues to increasingly replace overall survival as the primary endpoint in randomized phase 3 trials. And there are lots of reasons for this, some of them simply excuses. But it seems to me that we are asking less of a drug in terms of its impact by using progression-free survival. And at the same time, they're costing more and more to give. And so arising from this observation, and I will call it a bias, I have begun to do a little bit of personal work on the meaning of progression-free survival to patients and in value-based frameworks. And this work is being done together with Chris Booth, Mike Brundage, Andrew Robinson, Vishal Gayawali, and others. And the first question we thought was worth asking was, do patients know what progression-free survival means? And if a new treatment can prolong this, but does not have any impact on quality of life or overall survival, would they choose to take it? And how big an improvement in PFS would be necessary for them to trade it off in terms of the toxicity that the drug might achieve? So this performed some new research activity. And this article was just published summarizing the pilot work here. We did a pretty complicated little pilot study, which examined patient reviews following an education session and structured interviews about what progression-free survival meant and what improvement in PFS would be enough for them to take a treatment that didn't change survival at all. And about 85% of the patients actually would not trade increased toxicity for any length of delay in imaging progression. So you could say, what if it delayed it a year? Would you be willing to take the treatment then? 85% said no. And this raises some questions about the intrinsic value of asymptomatic imaging progression as an endpoint because patients seem to perceive it as low. This was a small study, and we've got CIHR funding to expand this to a larger sample across Canada. But the outcomes of this could have implications not just on clinical trial endpoints, but how people in the regulatory and reimbursement space evaluate new treatments given what cancer patients feel the meaning of this endpoint might be. The other things that happened at this point is I got involved with the Canadian Cancer Research Alliance. This was originally formed by Phil Branton and Vic Gling back in 2003 when the Canadian Strategy for Cancer Control was in development. They got together about 20 cancer research funding agencies to discuss what they could do together and share information on their investment. Now, as of today, there are now 33 research funding agencies involved in the Alliance, and collectively they invest about $500 million of research in Canada over the year. For almost a decade, I co-chaired the CCRA, which was then supported by the Canadian Partnership Against Cancer. And part of the work that we undertook together was to have a really honest discussion about how do we go from just telling each other what we're doing to really deciding what are the priorities we want to collaborate on and make shared investment in to accelerate impact. And that led to the creation of the Canadian Cancer Research Strategy, the first collaborative Pan-Canadian Cancer Research Strategy, which included 24 action items, one of which was to explore the struggling state of academic cancer trials. In 2010, we were hearing over and over that cancer centers and hospitals just couldn't afford to open an academic or a cooperative group trial anymore. They had to do pharma because they couldn't afford the costs. And 4 recommendations were made from this report on the state of cancer trials, including to create a national infrastructure program to support hospitals and clinics, to give them the funding to hire the data managers, to hire the ethics folks, so that they could participate in so-called academic trials. And this was formed in 2015, and we undertook international peer review and selected OICR as the home for this Canadian Cancer Trials Network, and Janet Dancy as scientific director. And it was really a collaborative effort. It took a lot of work to get 11 provincial, 2 national, and 1 project funding from industry funder together to underwrite the about 10 million a year that this was going to put into infrastructure and other supports. But it has been, I think, successful by many metrics, and I only show one here, and that is a 200% increase in enrollment in academic cancer trials in the last 5-6 years. So what about the last decade? Well, we are living through the days of the immune oncology revolution. Professionally, a lot of changes happened in my life that I'll talk about. And finally, lots of people are now all grown up. So I was going to call this the immune therapy wave, but it's more of a tsunami, really. And I can't go into the whole story, but it is really clear that starting with immune checkpoint inhibitors, CTLA-4 and PD-1, PD-L1 inhibitors, we've seen transformative changes to melanoma, activities that are changing practice in other cancers. And I know Francis wants me to mention lung, so I specifically will do that right now. We are also seeing the impact of engineering T cells in hematologic malignancies and research in solid tumors that will likely do the same. And the Canadian Cancer Trials Group, although I had left for some of this activity, has numerous clinical trials ongoing. I was actually back in the clinical trials group as acting director for a year when we were designing the adjuvant lung cancer trial, which I'm hoping will read out pretty soon with a PD-L1 inhibitor. We are doing an international trial looking at how long we should be treating patients with melanoma, a randomized trial with Australia, numerous combination studies, translational studies, and recently, Dr. Annette Hay, who works at the CCTG in Kingston, together with the collaborators across Canada, has got funding to develop a CAR T platform for research. Important to realize when I say tsunami that as of a year ago, there were 4,400 clinical trials ongoing of PD-1, PD-L1 inhibitors alone, and this is how they've grown. And there is no doubt that this is exciting, but there's some concern also to underscore here. Many of those trials are addressing either identical questions or variants of the same question with just an analog of the same compound, so that while this could confirm success, it will also undoubtedly duplicate failure, waste resource, and increase costs. I'm not sure what the solution to this is, but it is really going quite wild there. By 2012, as I said, I was thinking differently about things. We know that there were important gains in survival and reduction in mortality for some cancers, but 3 events changed how I was starting to think about cancer research. The first was I became head of the Department of Oncology at Queen's, and I was not immersed anymore in daily cancer treatment research. And this opened my eyes a bit to the reality of cancer system challenges. And 2 world congresses had illuminating events. The first was the 2012 World Cancer Congress UICC meeting in Montreal. And Chris Wilde, at this time director of IARC, said in his talk, the global cancer burden is and will become so large that we will not be able to treat our way out of the cancer problem. And this was like a little bit of a knife to the heart, because I'd spent 30 years trying to treat our way out of the cancer problem, with some success in some places, but I hadn't really taken this message home until that talk. And later that year, I got invited to the World Oncology Forum. And this was the first one of, I think, biannual meetings since that time in Switzerland, where 100 experts were brought together, asked one question, and put in rooms for a couple of days. And the question this time was, are we winning the war on cancer? And the answer was, not really. And the recommendations coming out of this were a 10-point action plan, the first one of which was prevent preventable cancers and wage war on tobacco. Now, this was the final published report, but what wasn't there was what some of us, including me, actually said should be that point, which was reduce smoking rates to 0%. And in that room at the time were all sorts of people who wanted to get rid of tobacco-associated cancers. And they said, we can't say this. We can't say this because we know it will never be possible. And that to me was rather shocking, because I thought if the people most vested in seeing this happen have already given up, how will we ever achieve this? And that led me to question, why the heck aren't we doing something about this? All of us involved in oncology, treating patients who are suffering from the effects of the single most preventable cause of cancer. Why aren't we speaking up? Why aren't we doing something? And so I decided to get a few people together and start to have some discussions. And is it possible for Canada to be a leader? And the long and the short of it was, this led to having a national summit in 2016 on creation of a tobacco endgame for Canada, where our goal was to achieve less than 5% tobacco use by 2035. We developed a background paper with 41 options and recommendations for measures that would get us there. And Queen's hosted this in September. And I will say that we are having a 5-year anniversary revisitation of the Endgame for Tobacco in a webinar that Rob Schwartz from the Ontario Tobacco Research Unit and the School of Public Health in Toronto is hosting on November 2nd, so tune in. We agreed to develop, we agreed on the need to develop an endgame strategy. The summary report is published online, and satisfactorily, a couple of months later, Health Canada, who had representatives at that meeting, identified this less than 5 by 35 as their goal for their new tobacco control strategy. So here's a lesson. I knew nothing really about tobacco control, but I got a little annoyed when it seemed that when we could make a difference, we weren't. And so my lesson here is applying learnings from leadership and research. Sometimes you can affect change in other areas that are of importance. The other thing that got me thinking is about the other end of what we do, and that's palliative care. Oncologists, especially people of my vintage, have a lot of experience with the need for optimal palliative care. And despite this, sometimes it's challenging to ensure that this is well integrated and available to all patients with cancer or other diseases. So this raised the question, are there different models that we could be using to integrate palliative care? And the fact that around the dinner table, I had a husband who was a family physician who did palliative medicine, and a daughter, Danielle, who was formerly at U of T on faculty, who's a palliative medicine physician. Those discussions sort of sparked that interest further. And we had a lot of fun, Danielle and I, in publishing this paper around evidence about overcoming barriers to implementation of early integration of palliative care. And that was our first paper together, and hopefully not our last. So what have we achieved in 35 years? Let's look at some numbers. The 5-year net survival for cancer is now 63%. Contrast that to 1947 when it was 33%. We can see from some of these net survival prediction graphs from this Canadian Cancer Statistics publication that we're seeing quite good figures for a number of cancers. And if we compare and contrast 87 to 2019, overall, we see quite dramatic improvements in 5-year net survival in many, although lung is still sadly not really knocking much above 20% yet. Now, some of you will argue, well, maybe that's all early detection and treatment didn't do anything. What about mortality? And when you look at mortality trends, you also see favorable trends in those same cancers over the time. And the CCTG has contributed to improved outcomes in ovary, breast, colon, and more. We've contributed to improvements, even though absolute small, in lung cancer and others, but there's still a long way to go for some of those cancers. And so our work is not done. So as I look back on 35 years at what was occupying our thoughts in terms of new drugs, there are definitely some hills and valleys there. Cytotoxics were how it all started. They're still around, but they've been replaced to a large degree by so-called targeted drugs and precision medicine. And there have been waves of varying sizes of taxanes, angiogenesis inhibitors, and now immunotherapy agents. And the size of that peak and the duration of that wave, I cannot yet predict. It's interesting to also say, what did we think was the root cause of cancer and what was going on over that period? Because in some way, the timeline of the drugs we studied reflected the evolution of our thinking about cancer. So when I started, it was all about cell division. The problem with cancer is uncontrolled cell division. If we disrupt the machinery of cell division or screw up the DNA, we will deal with it. And that's what those cytotoxic agents largely did. The idea that the DNA or the cells were dividing because they were being driven to by aberrant signaling pathways and mutated signaling molecules was the next sort of evolutionary understanding, and that led to the molecular targeted area. Then we started to even look outside the cell. It's not all about the cell. It's about what supports the tumor, its microenvironment, the angiogenesis inhibitors, and other potential therapeutic targets in that microenvironment. And now moving more towards whole organism, whole person, thinking about things like harnessing the immune system differently, looking at cell therapies, exploring what the microbiome might do to cancer risk and cancer treatment. At the same time, my professional interests have kind of evolved from clinical trials to thinking about research and professional organizations, coordination and strategy and research, cancer control policy, and finally, prevention, palliation, and now in my new role, innovation. So what comes next? Cancer biology and discovery research will continue to yield many new opportunities for therapy. We talk an awful lot about data, how to use it. We have mountains of it from patients, biospecimens, imaging, omics. How do we harness this, not to overwhelm us, but to understand better what are the predictive and prognostic indicators that it can reveal about patients and their disease, and also to design new treatment strategies and drugs. If I had to give advice to the next generation of clinical cancer researchers, I would say, be collaborative, enable us to achieve our goals more quickly. While early trials can start in one institution, eventually they need to move to multi-center studies to be able to understand the true impact of treatment on large numbers of patients. Resist the push to endlessly duplicate ongoing research. Ultimately, this costs resources of patience and time. And it would be far better to do fewer studies that were truly innovative than to just be busy doing things that we know are already ongoing elsewhere. I haven't spent much time on this, but for me, engaging patients as partners to understand what are the questions of importance to them, not just as research subjects, but truly as partners in designing the work that we do. And my advice, I doubt there are any of those on this call, to cancer research funding agencies and to government, is to invest in the academic side of clinical research by creating the infrastructure for healthy clinical trials community, not just for cancer, but for all therapeutic areas. We will be able to turn on a dime and conduct trials beyond those that are relevant to pharma. And all we need to do is look and see what was possible in the UK with their academic clinical infrastructure to, within 6 or 8 weeks of the pandemic, start to do large randomized trials of therapeutics for COVID-19 to show what that investment can bring. So 35 years later, we're all grown up now. I have many people to thank, especially the Canadian Cancer Society, whose charitable funds have continuously supported the Canadian Cancer Trials Group now since 1980, so for 41 years. And there are other funders shown there, all of whom have played an important role in the success of the group and our research. But also to the many, many mentors and colleagues involved in the Investigational New Drug Program and the thousands of patient volunteers. And on the side of my family, thanks especially to them for putting up with me and my travels and my babbling about cancer research over dinner. We're all grown older, for sure, grown up. Both my girls are in medical fields. My daughter, Nicole, a PhD in public health, and my daughter, Danielle, a palliative care specialist. And I have 2 lovely grandchildren. So much has changed since 1982, and I hope I gave you a sense of the times I've been privileged to live and work in. In addition to living through this time where we did see big impacts on cancer outcomes, many related to the trials of the Clinical Trials Group, I experienced a personal journey that was never planned or expected, and I have, as a result, experienced lifelong friendships and fantastic shared experiences and meaning in my work. But our work is far from done. Too many people are still developing, suffering with, and dying of cancer. So we have promises to keep and miles to go before we sleep. And those words, of course, come from this beautiful poem by Robert Frost. And with that, I thank you for your attention and welcome any questions.
Thank you, Elizabeth, very much for this absolutely sweeping panoramic view of cancer drug development and your own personal journey. We have actually run out of time, so I'd like to remind the audience to tune in to fill out the evaluations and the login. And I'm sorry we don't have more time. This was an absolutely inspiring and engaging talk. And again, congratulations, our deepest congratulations on your tremendous success and your award. So thank you very much. And thank you, everyone. I'm sorry we don't have more time, but would like to end and wish everybody a good afternoon. Elizabeth, thank you very much. Really appreciate it.
Thank you. And I'm sorry, I did time it at 48 minutes. So I guess
this was wonderful. It was a wonderful way of wrapping things up. Really appreciate it.
Thank you.
So thank you all. Take care.